23andMe's education program manager focused on the use of 23andMe as a learning tool for undergraduate and graduate classrooms. Previously, she ran her own coaching and mentoring business for students across all levels and entrepreneurs, especially female founders. She has a PhD in biomedical sciences. Dr. Taudot, welcome to 23andMe Live. You brought a science experiment. We got strawberries, a healthy snack for myself, and a special friend.
Yeah, I have a special guest here. His name is Arden Lee. He's in the 4th grader, and he'll help us out with a little classic experiment. This is a strawberry DNA extraction experiment. It's one of the ways that we get students excited about DNA and show them what DNA actually is. So I'll talk you through this, and Arden, you're going to help me out, right? So here we'll take some giant strawberries because they have lots of DNA and chromosome in it, and I just need you to take off the little green pieces and then put it in the Ziploc bag.
All of them?
Yeah. So let me help you out. I'll just take off the green pieces and put the strawberries in the Ziploc bag.
So any particular reason why we're using strawberries as opposed to another fruit?
Yeah, so strawberry has lots of chromosomes, 8 sets of chromosomes typically, and that's why when they breed the strawberries, they make it so that it can be nice and extra big.
Sure.
Yeah, in comparison, humans only have 2 sets of chromosomes. So we're just going to squeeze out the air a little bit so that when you...
Which would have been awesome, but probably a disaster for wardrobe. So.
Yeah, we'll be wearing the strawberry on our shirt. And then I like to put it in another Ziploc bag and just seal it. So basically what we're doing is every human being, living things, animal, plants are made of millions and millions of cells. So here I have an example. So this is animal, this is a plant cell actually, and then this is an animal cell.
Great.
And so what Arden's going to do is, I just need you to completely destroy it. So what we're doing is we're breaking apart all these cells and pulling it away from each other. So at the same time, Mike, if you can help me make a lysis buffer or extraction liquid. So what you're going to do is pour half a cup of water in that cup. So what we're going to do is create a little solution, a little buffer that will pop the outside walls of the cell and allow the nucleus and the organelles and other parts of the cells to come out and fall out.
Which is part of the shakeup right now that Arden is taking part of.
Exactly
Okay. Now I have all this stuff is stuff that I can go to the store and grab. I mean, you mentioned the water. I got dishwashing liquid here as well. Am I supposed to be pouring some of this dishwashing liquid just now?
Yeah, so 2 teaspoons of dishwashing liquid. Doesn't have to be exact. It's a fun science experiment, and that's the fun thing about this experiment. You can do it at home. You can do it in a science classroom, in science fair conferences. You can do it with people of all ages, so it's super fun. And then just 1 teaspoon of salt. 1
teaspoon of salt. I feel like Arden might have gotten the cooler job because I'm doing all the he's got all the hard labor. I guess mine is the
salt a little bit.
Yeah, as you can tell, don't spend too much time in the kitchen. So.
And you can use any salts.
And you said 2 teaspoons?
Just 1 teaspoon will be good.
Okay.
Yeah, and with dishwashing liquid, I like to use the clear dishwashing liquid, but any soap will do.
Great. Now, Arden, how do you feel right now having fun with the strawberries?
It's hard because
smashing strawberries is hard?
There's little chunks that are small and like slippery.
Yeah.
The amount of diligence that Arden is now displaying, making sure that every single chunk is taken care of, is very impressive.
Yeah, and this is why it's great to do with kids too. So you just want to stir your solution a little bit with your salt and your dishwashing liquid. So what's going to happen is the dishwashing liquid, it's the type of detergent, and when it comes in contact with the cell, it'll pop holes in the cells.
Great.
And so imagine if a cell is like a water balloon with a bunch of stuff in it. The dishwashing liquid will pop holes and make the cells open up.
Perfect.
So all the DNA and nucleus will fall out. So now you can just add a little bit of that liquid into this bag of strawberry juice that Arden has created.
All right, you did an awesome job with this, and I am worried that this strawberry jam is going to be all over. So now, am I pouring?
You're just going to pour a little bit in there.
Well, hold on one second. I got to pour some of this in here first.
Yeah.
So
there we go.
Is there a certain amount or just kind of?
Just a little bit, a splash.
Is
That's
that good?
Yes.
Okay.
Like making a cocktail.
Arden, what do you know about making cocktails?
Nothing.
Okay,
I know how to make PB&J.
Good. That's,
Is that the same thing?
It almost is, and
Now
clearly
you're
you
just
can make
kinda
your own
like
jam.
shaking it a little bit, just get the juice to come and mix it around. And that'll allow the detergent to work into the DNA.
So
in the
what exactly,
strawberries.
like I took water and dishwashing liquid and some salt, and we just mixed it with strawberries. So what am I supposed to be looking for right now?
So right now, what's happening at the microscopic level, where you can't even see it, is the detergent is popping the holes outside the cell membrane.
Sure.
And all these components is falling out now, just coming apart. And you're basically destroying the cell. And that's why we use dishwashing liquid to clean stuff, because that's what kills bacteria. It just pops the cell wall and makes things fall apart.
So what are we trying to isolate? If that's the cell and that's
right
what the
here.
strawberry is.
So inside almost every cell, there's a little nucleus, and the nucleus contains DNA. So we're making this fall apart. We're making all these components fall apart, and now we're going to get you to help us filter
great.
it out.
So I am now going to put this coffee filter in. And how about Arden, you help me out just a smidge with this. We're going to pour some of this into this coffee filter.
And you can use coffee filter, or you can use paper towel, anything you have flying around, cheesecloth. It's kind of fun too. And what you're doing is just basically, getting the bigger chunks to like isolate. And the DNA is super tiny, so it's going to fall through this filter.
What am I trying? What's the? Obviously, we're trying to separate the liquid from some of the chunks that are in here, and obviously, I'm doing an awful job of that. But
there we go.
Some of it's pouring through. What's the end result? What are we looking for?
Some of the DNA is falling through it, because the DNA is super tiny. It'll fall through the filter.
So there's DNA coming off of this coffee filter from that strawberry.
Exactly.
Because I think people think about DNA and it's something that you can't actually see.
Right, and they think it's like some mysterious thing. So we're gonna unmysteriousness of it and demystify it. Yeah. So just squeeze a little more, so we
you
can
want me
get
to
like
squeeze through here?
Yeah.
Okay. I'm making a mess here, but I'm doing okay with the.
Yeah, you're doing pretty good.
Is there a certain amount of juice that we're looking to?
Just a little bit, so we pour in this little cup or in this test tube.
How is that? That doesn't look like it's enough, right?
Yeah, we'll probably need a little more. But
Okay, I'm gonna get real dirty and just we're just like squeezing lemons here.
And this is like the great thing about doing experiments with kids.
I feel like
you just have a little fun with it.
Dr. Tao, I know you assigned us tasks. I feel like Arden might have been a little bit better with creating the juice. I might have been a stronger smasher of strawberries, getting some of that anger and frustration out.
Okay, so
is that
cool.
enough?
Yeah.
Okay, I'm gonna just slide this here. Is that cool?
Yeah, you can drop it in here.
All right. Well,
all right. So we'll pour a little bit into this test tube.
It's not my job, by the way, to clean up when we're done, which is probably the best part about this. How much?
Yeah, just
is that good?
That's good.
Okay.
And then you can put the rest in this cup here. And then what I'm going to do is I'm going to use rubbing alcohol, which you can use ethanol, isopropanol. Usually 95 to 100% is good. People like to say that you can put it in the fridge first.
Okay.
But you don't have to. So I'm just going to pour it in here, help Arden out. Obviously, alcohol doesn't smell very good, so don't smell it and drink it. But what I'm going to do is just lean the test tube and then pour a little bit of alcohol and create like, oops.
Arden, have you done anything like this before?
Ish. I've touched rubbing alcohol before, but I haven't extracted DNA from strawberry.
Well, look, this is it's my first time as well. We are learning together. And I love how my hands were the ones that ended up dirty. If you would have told me that would have happened between a kid in 4th grade or an actual adult, and maybe that's the reason. My maturity level probably the same as Arden's here.
Well, I'm a 6th grader at heart too. So here, we can use a clear cup if you have it at home, or you can get a test tube. Like if you're in a classroom and you have 60 kids, you can buy a 60 test tube for $10. It's super cheap. So you can get the kids to feel like a scientist, and it's completely affordable.
We need to get you a lab coat next time you come out here and smash, so you really get, you'll look the part as well. But as I'm seeing you pour some of that alcohol into the cup and the test tube, there's some bubble action going, and there looks like some white substance that's in there.
Yeah, what do you think that is, Arden?
It's gucky and it's kind of slimy. I have no clue what it is.
Yeah, what do you think it is?
What do you think?
To be honest with you, if that was coming out of your nose, I'd tell you that you need a tissue to wipe it.
It looks like a little
It
snot.
kind of looks like snot, yeah.
Yeah, so you can take a little coffee stir and try
Okay.
to like pull it up and look at it. So this is strawberry DNA that you can see and feel. So what happened is when I pour the alcohol on top, the DNA comes out of the solution and just become this nice little thick solid. So you can see and touch it.
So that is actual DNA is what you're telling me.
That is strawberry DNA. And so if you look at the DNA, it'll look kind of like this. So here's a 3D model of a DNA strand. So inside each cell, there's a DNA strand this long. It's about 6 feet long,
Great.
actually. And it gets twisted up so that it can fit inside a tiny little cell. And inside every cell has DNA. And that's basically what 23andMe analyzes. We look at the structure and we see what it tells us about ourselves.
Any idea why there's a spiral to the DNA?
Could be just naturally. I don't know.
Yeah, so the DNA gets twisted up so that it can like fold itself and be super organized inside like these tiny little cells.
All right.
Yeah, and you can see it here too in a test tube. You can see the 2 layers. So that's why it's great to use strawberry as well. The red color and the clear color is really nice to see. But you can see the strand of DNA just kind of twisting around. And it kind of floats on top.
Well, fantastic stuff by you guys. We've had some pro athletes stop by the show with us, but I got to be honest with you, Arden, I think you might have stolen the show with the strawberry smashing and holding snot with a coffee stir. So job well done by you.
Thank you.
Dr. Tao, thank you so much for
Yeah.
the insight. Really do appreciate it. We got plenty more. We're coming back with more 23andMe Live. That's straight ahead.